All right, let's get started. Last week, I apologize, I forgot to attach the PowerPoint so for today, so we didn't get that up till this morning, so sorry about that. But I keep doing that wrong. I think I've got it for the end of the semester though. I got everything posted that we're gonna do. I think we're gonna have plenty of time getting through the 4 pathogens that we have left. I thought about, I think we'll try to push through today and get through as much as we can. I still want to do some review questions with you today. If we should have a little extra time on Thursday, we can do a little bit more reviewing. So we'll go ahead and kind of just push through and get done what we can today. But I did want to do a few review questions here just from, remember, this is from our most recent stuff since the last exam. So I would encourage you to spend most of your efforts studying on this most recent stuff material because you're most likely to see that on the exam. Definitely, if you have time, go back and review stuff that we did in September and October. But again, you're not going to get the bang for your study hours because you're just kind of less likely to actually study the right thing, I guess. So again, especially be familiar with what we've done so far. So maybe you had a chance to look at this already, decide what you think. So strep pyogenes, which of the following is false? This question gets a little bit more specific. Like I wouldn't ask you what Lancefield group strep pyogenes is, or I wouldn't ask you its hemolysis type. Those are maybe lab questions. These are both actually true. It is beta hemolytic and it is has group A Lancefield antigens. More likely, okay, what is a Lancefield antigen? These are just carbohydrates on the surface of Streptococcus. So it's a way that we use to kind of put Streptococcus in groups. It produces streptolysins, right? Those are exoenzymes. And this is true as well. It can be lysogenized by a temperate bacteriophage. So it can kind of, if that organism is infected by a virus, it can actually change its phenotype and be able to produce an erythrogenic toxin. So there are strains of strep pyogenes that cause this red rash. All right, so the one that is not true is A, that's actually Staph aureus we talked about that can make protein A. So the next question follows up there, untreated streptococcal pharyngitis, strep throat, that can progress to which of the following? All right, give you a second to read that. You can chalk it over if you want. Not chickenpox. We're going to talk about that today. That's viral. Okay, and hopefully you don't confuse strep throat with anthrax. Remember, that was Bacillus anthracis. And necrotizing fasciitis, that is actually also caused by strep pyogenes, but that's pretty much one of the worst things you can get. We don't talk about complications from that. That would be a complication. So both scarlet fever or rheumatic fever. I really don't like those questions where you have to kind of read to the end. I mean, I hope that you look at all the answers, but you might just, circle that one and then move on. The other complication is that acute glomerulonephritis, where it's kind of another autoimmune complication of an untreated strep throat. A couple more. Okay, anthrax question. I did not know this till I came across this question. Again, not one that I wrote myself. Anthrax, which means charcoal in Greek, derives its name from what? All right, I'll give you a minute to read over that. So I like this question because all these things are true. It's a good review of anthrax. All right. Remember that what's unique about that infection is that the Bacillus anthracis is a spore former. Okay, it makes endospore. Well, this one doesn't really make sense. When we stain it under the microscope, it doesn't stain black unless we use a black stain. So that doesn't really make sense. Correct answer here is C. The black eschars it produces on human skin. So that's a good term there to be familiar with, those black scabs that the patient can get from cutaneous anthrax. This is true to some extent. It's the toxins really that invade the bloodstream. Definitely high mortality causes in infected individuals, but okay, answer C there. One more about anthrax. So which of those is false? Okay, so remember there's three ways you can get anthrax. Through a wound, you can breathe in the spore, you get the spores get in a wound, you can breathe in the spores, or you can ingest it and eat the spores. Let's start at the bottom here. Yes, it can be lethal even after treatment because there's a couple of toxins that are working. So the antibiotics are not going to be working against the toxin. Yes, it normally dwells in the soil. Okay, those endospores can be viable upwards of thousands of years possibly. It does have a capsule. We didn't talk about that a whole lot, other than to mention it was kind of a capsule that is a little bit unique. It's more of a protein capsule rather than polysaccharide capsule. It does produce endospores. So correct answer here, this is a zoonosis. So humans only get anthrax when they are in contact with animals. Humans don't pass it to other humans, right? So primarily a disease in animals. We only kind of incidentally get it when we're in close association with animals. All right, so that was a little review of the streptococcus, the anthrax. Last lecture, we were talking about the gram-negative cocci, the Neisseria gonorrhea, Neisseria meningitidis, and talked through those two infections. So if we have time, we'll do some questions to do with those infections after we go through our DNA viruses. Let's see, what else did I want to say? Okay, so we'll have some more review there. Let's go on. Chapter 24 is our DNA virus chapter. Remember, you can be working on the chapter 19, the chapter 20 homework. Again, I don't think it's too extensive this time, so that shouldn't be too much work for you. We're only going to pick out one viral pathogen from chapter 24, and that's herpes virus. It's kind of a complicated virus. There's a lot of different a lot of different variations of herpes virus. So we'll get into that. Just to review a minute about viruses in general. So we talked about viruses quite a bit in relation to our immunology chapter. Remember that viruses can only reproduce inside a living cell. So on your last exam, there was a question that was a little bit of a curveball. It said, I think, why can't viruses infect our outer layer of skin, or I don't remember quite how it was phrased, but the question had to do with our outer layer of skin is dead. All right. Remember, viruses need a live cell in order to infect. So you don't hear about having viral skin infections, right? That's just not really a thing. Warts actually will penetrate through cracks in the skin. So as long as the skin is intact, then you wouldn't get that. So again, they need a living cell in order to reproduce. Remember, virions are composed of some kind of nucleic acid, either DNA or RNA. All right, as far as we know, there's not a virus that has both. You'll have one or the other. And then that nucleic acid is going to be protected by a protein capsid, all right, made of capsomeres, individual proteins. All right, and so we're interested in viral pathogens. So we're thinking about viruses that infect animals specifically, not viruses that infect bacteria, not bacteriophage. Most viruses that infect animals are enveloped. There are lots of exceptions to that, but generally speaking, that's the case. So again, most of the viruses that infect us would have this phospholipid membrane around it, which we call the envelope. All right, so, and remember, I guess I just put this up, this kind of talks about how we classify viruses a little bit. We had one scheme that was just based on the nucleic acid, the Baltimore scheme that was 7 different categories. A lot of times though, we try to make viruses fit into our classification system like they were alive. So we have families and there's a few orders and families and genera. Viruses are usually just named for the disease. Okay. So just a little variety there, nothing to memorize there. Let's look at herpes, the family Herpes Viridae, kind of generally speaking, and then we're going to look at some of the different types of herpes virus and the infections that they cause. All right, so in general, Herpes Viridae, this is an enveloped viruses. They have a capsid that's icosahedral. So remember, icosahedron, that's a 20-sided polygon, a very stable structure, lots of strength. So you can see that over there in the picture there, icosahedral capsid. It has linear double-stranded DNA as its nucleic acid. Remember, there are viruses that have single-stranded DNA. It's not something we ever find in any other organism. So what I mean by linear is that their DNA is all in one strand. Okay, some viruses, like we'll see, like the influenza virus has lots of different segments. Okay, this is all in one strand, and it's a pretty big genome. We think it encodes for somewhere between 80 and 100 proteins. Okay, so that's pretty large. Smallest virus, remember, only has about 4 genes. So 80 to 100 genes that encode for, about that many proteins. What's fairly unique about Herpes Viridae, there are a few other viruses that have a tegument as well, but Herpes Viridae have a very pronounced tegument, and that's drawn in purple over here. So all these are proteins that line the inside of the membrane, the envelope, that the virus carries with it. We call these tegument proteins. We think that there's somewhere around 20 tegument proteins, and those just help the virus be successful. So sometimes some of them aid in viral replication, some of them aid in entry into the host cell, some of them act to suppress host immunity. For example, there are some that downregulate MHC class 1. So think about it, when the virus is inside a host cell, that's the only way the host has to recognize that that cell is infected when it's sampling with MHC class 1 and presenting that antigen on the surface. So very sneaky of the virus, and there are other viruses that can do this too, to download that presentation by MHC class 1. So it's going to be more successful hiding out in cells, less likely to be noticed by our cytotoxic T cells. Another prominent feature, many viruses have them, they're surface glycoprotein spikes. So we're not going to really name these too carefully for Herpes viridae, but they're essential to recognize, bind to, and help the virion actually fuse with the host cell membrane. Most of us have been infected with some form of herpes virus. So it's very widespread. Sometimes if you were infected when you were young, perhaps your immune system fought it off and it didn't go latent, but probably about half of us have herpes viridae latent in our cells. Reservoir is humans. So animals have herpes viruses. They don't typically cross species barrier. There's one exception to that. Apparently there is a herpes virus in monkeys that very rarely, but sometimes will infect humans. So it seems like there's an exception to everything in biology, but for the most part, again, they kind of, again, humans, we pass it around to other humans. Let's see. All members show latency. So that means once you have a herpes infection, and again, occasionally we think that people fight it off and it doesn't go latent, but a lot of the times it does go latent. So it goes latent in a little different form. So remember, bacteriophage that can do lysogeny, that means that they can incorporate into the bacterial chromosome. And when they do that, we call that a prophage. When animal viruses incorporate into the host chromosome, we call it latency. So lysogeny in bacteria, latency when animal hosts, and we would call that a provirus when the virus incorporates into an animal cell chromosome. Herpes virus has a little different strategy. They actually form episomes instead of incorporating into host DNA. So we could, let's see, these are plasmid-like structures of circular DNA. Okay, so here's a host cell. Maybe I could, here's the DNA in the nucleus here. And the herpes, instead of incorporating into that DNA that I drew as red, will actually in the nucleus just kind of have this little ring of the herpes virus DNA. And once it's there as an episome in the host, this is going to be permanent. Unfortunately, we haven't figured out how to get rid of herpes virus once it's gone latent. So it just kind of divides whenever the host DNA is going to divide. Some herpes are actually oncogenic, meaning they have the potential to be cancerous. And we'll talk about one of those in particular, Epstein-Barr virus. It seems like environmental factors, predispositions kind of play a role there as to whether that is successful. I just posted this link. That's just like a 2-minute video. If I had a way to easily jump to that, I would do that, but I don't always do that successfully. It's just a 2-minute animation of kind of the life cycle of the virion when it enters a host cell. it has to it has to enter the nucleus. So DNA viruses usually will enter the host nucleus. That's where the enzymes are that it needs to replicate its DNA. It actually will pull the nuclear membrane with it instead of the cytoplasmic membrane. So that's kind of unique about herpes virus, but it's a great little 2-minute animation that kind of helps you visualize what's really going on there. So I'd really recommend that. Little more general characteristics. These viruses target mucosal membranes, and they multiply in basal and epithelial cells. So basal cells are kind of the deepest layer of the skin. Those are the cells that make our skin cells. They typically multiply there, and then they'll head to our nerve cells, causing a lot of inflammation, cell death of infected cells. How do we diagnose herpes virus? We don't just take a sample and look for them under an electron microscope, right? We can't see them under light microscopy, but to look for them with electron microscope is a lot of time and expense. What we typically do, somebody has symptoms, we take a sample of cells and look for a particular cytopathic effect. Okay, we might abbreviate that CPE, cytopathic effect. Okay, and so a cytopathic effect, that means a change in host cells due to the virus. So that's going to be visible with light microscopy. Okay, so a particular cytopathic effect that we look for is the formation of syncytia. Let me make sure I spell that right. Syncytia. And what syncytia are, these are multinucleated cell. Let me write that again. All right, so here's a couple of examples of multinucleated cells. This is actually a really ingenious strategy by herpes virus, because if it is going to enter a cell and then, lyse or bud off some way from the cell, it's going to be vulnerable for a time when it's outside of the cell, vulnerable to things like antibodies or phagocytosis. So what it does instead, one infected cell will just fuse with the neighboring cell that's not infected. So in that way, the virus doesn't have to go extracellular, and it's not, again, vulnerable to antibodies and macrophages and things like that. So here's a nerve cell. All these little purple dots here, these are nuclei. So that's a cell that's actually fused with lots of other ones. Over here in this cell here, you can see the multi-nuclei here as well. So there are other viruses that will cause syncytia. It's a very prominent thing for herpes virus. So again, host has symptoms and you see that, and that's an easy way to diagnose. All right, so let's look more closely at some of the different herpes virus that are out there. There are actually 9. So when I looked this up yesterday, I was like, oh, I need to update this. But the 9th one is just the one that's usually in monkeys and only very rarely infects humans. So we'll stick with this. We used to call it herpes simplex virus. At some points, I don't know, they changed the name to human herpes virus. So HHV instead of HSV. We're going to look a little bit more closely at the first 4 of them. So HHV1, that's sometimes referred to as simplex virus. That's typically going to cause fever blisters or cold sores. Okay, you could think of it as above the waist herpes. HHV2, also called simplex virus. This is usually genital herpes. genital sores, or below the waist herpes. Sometimes it's possible to get Hhv2 sores in your mouth, but typically that's due to oral sex practices. Hhv3 varicella virus, that's a causative agent of chickenpox, as well as shingles. Talk more about that. And Hhv4 Lymphocryptovirus is more commonly called Epstein-Barr virus, after the people that discovered it. And that's most commonly causes infectious mononucleosis. All right, so the other 5 I'll just mention here, but we're not going to talk about any more detail. Cytomegalovirus, most people haven't been infected with that at some point in their life, and oftentimes that doesn't cause any symptoms. It's more a problem if you are immunocompromised. Apparently, about 10% of mono cases are actually caused by Hhv5. 6 and 7 cause roseola, which is usually kids get when they're very young. It causes kind of a real flushing or redness of the cheeks and high fever for a couple days, but then usually resolves itself. And Hhv8 retinosvirus causes Kaposi sarcoma associated cancer. That virus actually suppresses, it actually acts on a tumor suppression mechanism that we have. So patients with this get a lot of tumors, but it's only people that have a very depressed immune system. So typically we find that associated with AIDS patients. All right, so that's just a little overview of the different, I guess we most correctly call these genuses of herpes virus. Let's start with Hhv1. Look at how that manifests itself, some symptoms. So gingivostomatitis, again, that's most commonly, we would just call that cold sores. Again, about half of us maybe will get a cold sore from now and again. Painful vesicles on the outside of the mouth, sometimes a little bit on the inside of the lip as well. Usually have a little bit of maybe prodromal period, you know, you feel a little tingling before one comes out. That's going to be transmitted by close contact. Okay, so usually you have to come in direct contact with a lesion, kissing, for example. Could be a shared drinking glass by some fomite, or maybe an eating utensil. Usually a cold sore will last somewhere between 7 and 10 days. Okay, so kind of annoying more than anything else. We can't actually get Hhv1 infections on other sites besides the mouth. If we get sores on the fingers, like right here, we would call that Whitlow's disease. That's the so the virus is going to enter. How common is it to have, you know, little cuts in your cuticle there? So the virus enters through little breaks in the skin and then, you know, manifests in little sores. That's one of the many reasons that dental hygienists wear gloves and dentists wear gloves. It can actually infect the eyes as well. We call that herpetic keratitis. It causes a real light sensitivity, can be serious, can lead to vision loss. So that can be kind of dangerous, and it has the potential to infect the brain and spinal cord. Typically, that's not unless there's been some kind of trauma, and typically that's only in immunocompromised patients. But there's substantial mortality even with just Hhv1 when it does infect there. Okay, Hhv4. I spared you any graphic pictures here, but this is genital herpes, fairly common. It's estimated that about 1 out of 6 people have genital herpes, usually contracted as a teenager, young adults, when people have, as a result of sexual activity. So it enters through tiny breaks in the urogenital tract during sexual activity. And people that are infected may have outbreaks maybe 2 to 3 times a year, most commonly. But some people have them as often as every 2 or 3 weeks. So it just kind of depends. It can be spread even when there are no active lesions, although less commonly when the lesions are not out. Causes itching, sometimes some discharge, sometimes fever. Okay, so there's sores that, again, are itchy, little discharge coming from them. Just like gonorrhea, we have to be very careful to try not to infect babies born to infected mothers. So if the mother is having an outbreak at the time of delivery, they will usually deliver the baby by C-section rather than vaginally. The babies most often would get a infection of the eyes that kind of resembles the gonorrhea infection in the eyes, but it has the potential to move to the central nervous system to the baby and be very, very serious. So again, try to avoid that by delivering by C-section. So both Hhv1 and Hhv2 can be latent. Okay, what kind of cells are they latent in? They will retreat to nerve cells. Hhv1 fever blisters are usually going to follow either the mandibular, maxillary, or oculary branch of the trigeminal nerve and be latent in that trigeminal nerve. So every now and then they'll just reactivate and you'll get the sores come out. Whitlow would most likely go latent in the brachial ganglia, and Hhv2 would more likely go latent in the sacral ganglia. What causes it to reactivate and start replicating again? Sometimes we don't know. Usually it will have something to do with some kind of stress. So just being outside on a sunny day could be considered stress. Menstruation, some kind of immunosuppression. So maybe you're on a steroid drug for a time and that causes a reactivation. Certainly having cancer, going through cancer treatments, or maybe just being sick with the flu or having some kind of a fever. My husband had not had a cold sore in over 25 years. And this summer he got one for the first time after he had his second COVID shot. So I was like, oh man, he had it really bad. He just was miserable for a couple days with that. So I guess it's not too surprising, but we just didn't think of that happening. Usually whenever it's reactivated, usually the outbreaks are less severe. So over time, they're not as often and maybe not as uncomfortable. So what can we do to treat this? Like I said, it's latent. We don't have any way to get rid of it, unfortunately. Really, our best bet is a drug called acyclovir. And my graphic here makes it look like it's injected, but I think you can administer it in lots of different forms. You can take it orally. There are acyclovir creams as well. This is a nucleoside analog. So we talked about these compounds back when we talked in chapter 7 about different mutagens. And there was a little confusion there. A nucleoside analog is a compound that looks very similar to a nucleoside. So a nucleotide is deoxyribose, the base, and the phosphates. A nucleoside is deoxyribose and a base. In particular here, guanine. So acyclovir, it's the guanine base and deoxyribose. So the fancy name for that is deoxyguanosine. That's the guanine nucleoside. Acyclovir then has the base that looks just like guanine, but you can see here deoxyribose is not really anything close to deoxyribose. It has no 3' hydroxyl group. Remember, that's where the new nucleotides or the triphosphate nucleotides would add on in order to make new DNA. So basically, because there's no 3' hydroxyl group, it terminates replication. Okay, so usually viral polymerases are kind of indiscriminate in terms of what they incorporate into their DNA. So the viruses will incorporate something that just kind of looks like a nucleoside into the DNA very easily, doesn't have really error checking mechanisms. And so that gets incorporated, and then it can't add on the next one because there's no 3' hydroxyl group. So causes chain termination, so slows down replication of the virus and hopefully reduces the length of the outbreak. So we do have acyclovir. For cold sores, usually just something over the counter, some kind of lip ointment that has menthol or camphor in it, just to kind of try to reduce the irritation. There is, I kind of check on this every now and then, there is a herpes vaccine. I don't believe it's reached clinical trials yet. It's a vaccine that's not given to you before you get herpes. It's given to a patient after they already have herpes. And it's supposed to reduce the length of the outbreaks and make them less common. That's kind of been in the works for a long time, so I don't know what kind of time frame we're looking at there. And I forget what kind of vaccine it is too, so not a lot of help. Oh, a live attenuated vaccine. Okay, it's supposed to decrease viral shedding as well. The WHO, so the World Health Organization estimates that 500 million people worldwide have Hhv2. So there's a lot of it out there. A little bit less serious generally is Hhv3, varicella zoster virus. Zoster is Greek for belt. And that seems odd when we talk about chickenpox, but I'll come back to that in a minute. Again, this is the causative agent of chickenpox. Let's talk about, we'll talk about what the symptoms are in a minute, but let's first talk about transmission, because it's quite interesting for this virus. It's humans only, despite the name chickenpox. Sounds like it came from a chicken. I think it was named that because somebody thought it looked, the rash of the skin looked like a plucked chicken. I don't know. I've never plucked a chicken, so I can't really say. But that's why it's called chickenpox. Humans only, passing it around with each other. It's passed by respiratory droplets. So usually have to be pretty close contact to somebody, usually within 6 meters. So it's this big respiratory droplets from, coughing or sneezing. The incubation period. So remember, the incubation period is before a host has symptoms, is somewhere, it's about 2 days. I've read in places that it's longer than that, but we'll go with the 2 days. The host is going to be most infectious in the incubation period. And is still infectious actually until the rash scabs over. So we'll say through scab formation. So that's about a week. But they're starting to be infectious in that incubation period when they don't even have symptoms. So this really adds to the transmissibility of chickenpox because people are out not even knowing that they're sick and passing it on. They're shedding lots of virus and giving it to other people. So this is a good opportunity to talk a little epidemiology. When we're talking about transmission, we often refer to a term called R0. Okay, so you pronounce that little zero as not, R0. And what this is a calculation. Let me define this right. This is a calculation of the number of people a sick person will infect. If you assume that the entire population is vulnerable to the virus. So maybe we could have just typed that out. That would have been easier. Okay, so this relates to the pandemic that we're in as well. We've been fighting about, oh, what's the R0 for coronavirus for a long time. Well, let's compare that, the R0 of several different infections. So again, this is the number of people. If you're sick with chickenpox, what's the number of people that you're likely to infect if you assume that everybody can get it? So assume that everybody has not had chickenpox or has not had the vaccine. They usually put that R0 for chickenpox at about 10. So pretty infectious. That's part of the reason before vaccination, almost every kid got chickenpox. And once you get chickenpox, you usually don't get it again. So that a good memory to it. I've spoke several times that measles is probably the most infectious disease we have. They put the RNA at measles, at least in this article that I cited down here from the CDC, at 18. All right, so that's why it's so important to keep our measles vaccination up, because when that lapses, if you're not vaccinated, you're likely to come into contact with somebody at some point that will give it to you. So just, you know, for kind of reference sake, again, like I said, they've been fighting about this for a long time. How infectious is coronavirus? This particular article puts the Delta variant at about 7. So originally they had said, oh, it's as infectious as chickenpox. Maybe not quite that much, but somewhere around 6 or 7 people. So, you know, if you're around, again, everybody that's not vaccinated or doesn't have any immunity to it, you're likely to infect 6 to 7 other people. The original strain was probably about 3. So just slightly more infectious than influenza. Anytime the R0 of a disease is above 1, then it's spreading in the population. So just kind of for reference, thinking about, you know, public health measures and kind of what we need to do to try to keep things from spreading. Symptoms of chickenpox. Probably there's been some of you that have had this, but probably not very many of you. That I don't skip anything here. Rash. So it usually starts out as a rash. Usually feeling fine until that rash breaks out. And it usually starts on the trunk, kind of the warmest part of your body, and moves out from that. I can remember when I was 8, waking up one morning and my mom took me in the bathroom to look in the mirror. I just cried because I had spots all over my face, and I don't know, I felt bad. So accompanying that rash are things that you might get from any viral infection, things like fever, malaise, loss of appetite. So that rash are these little small fluid-filled vesicles. And once they start to scab over, they get very, very itchy. So again, very intensely itchy. So usually chickenpox clears up on its own. Not, when kids get this is not a usually very serious infection. There's kind of a low chance of complication. Once you get it once, you usually don't get it again. Sometimes you hear stories, oh, so-and-so got it twice. Well, they maybe had a really mild case and didn't establish good memory for some reason. So when the vaccine to chickenpox came out in the I think it was 1995, there was some pushback to this because people argued, everybody gets this, it's very mild. Why do we need a vaccine, right? There's always, risks anytime you take any drug or vaccine of side effects or allergic reactions or whatever. But I want to present with you 3 really good reasons that getting vaccinated to chickenpox is a good idea. So I'll start with kind of the, weakest argument and then move to the strongest. So why do we want to vaccinate? Well, there is the chance of secondary infections. That rash, when it scabs over, it gets so itchy. You almost always are gonna, break some of those scabs off before they're ready to fall off, and you're gonna have, some portals of entry there in the skin. So there's potential to get some kind of staph skin infection. Again, we have antibiotics that maybe not is too big a deal. There is the potential for some more serious complications like pneumonia or encephalitis, which is inflammation of the brain. Pretty rare though, those serious consequences when kids get it. I think a stronger argument is that, remember, once somebody is infected with chickenpox, it will go latent. And it can go latent really in the trigeminal, brachial, or sacral ganglia. And at some point later in life, if somebody had chickenpox as a child, that can reactivate later in life as shingles. And about 1 out of 3 people with chickenpox will get shingles. Usually it's later in life when their immunity maybe starts to wane a little bit. I've talked to a high school kid that had just recovered from shingles. So that's not always the case. I've had friends in their 40s that got shingles, but that's usually the case, usually when they're older. And again, about 1 out of 3 of us. So what does shingles look like? It's a very painful rash. So again, not itchy this time, painful. And this is where that zoster, that belt name comes in. Because shingles often will look like, this swatch right around the belt area. Not always, but that's one common place for it. It's interesting, it's almost always one side of the body or the other. This picture here, you can see that sidedness, it's really striking there. So there it's reactivated in the trigeminal nerve, and you got this painful rash on the face. That looks just really nasty. And that usually will clear up after maybe a week or 2. But about 10% of patients that get shingles, the pain of that will just go on and on and on for months sometimes, and they call that post-herpetic neuralgia. So again, that's just, you know, the pain lasts for sometimes months. Again, that's only about 10% of people that have shingles, but a third of people get it. Still a pretty high percentage, and then we don't really have much that can help with that. We try a little acyclovir, but that doesn't always help very much. And somebody that has shingles, so you can't give shingles to somebody else, but somebody with shingles could give somebody the chickenpox if they hadn't already had it or been vaccinated. So, you know, I think there's a picture in your book of a grandpa holding baby twins and says, oh, he's had shingles. Okay, well, he could give chickenpox to these babies, which could be dangerous to have, you know, any infection in babies. So keep that in mind. I had a student that said, I work in a nursing home and I never had chickenpox or was never vaccinated. Should I get vaccinated? I said, yeah, you should probably go today to get vaccinated, because when you get chickenpox, when you're a teenager or an adult, it can be much more dangerous. So this is really the strongest factor to get vaccinated. Age is a factor. Again, teenagers or adults that contract chickenpox are going to have a much higher chance of some kind of complication. Some of you may know Dr. Baxter. He's in the BMS department. He had chickenpox as a teenager and was sick for a really long time. He had pneumonia from it. So, you know, if you don't get it as a kid, you really should get vaccinated. Just we just have a lot more inflammation that's involved when you get it as you're older. All right, questions so far about chickenpox? All right, vaccine. What does a vaccine look like if that's so important to get? So the chickenpox vaccine is called Varivax. Oops. Chickenpox. It's an attenuated vaccine. So live attenuated, weakened, somehow they've weakened the virus. It's usually given in 2 doses, about 18 months, and then again at usually, you know, before kindergarten, 4 or 5 years. And it works really well. It's very effective. And if you've had the vaccine and you've never had chickenpox, you should never have to worry about shingles. So that's definitely a plus. People like me that had chickenpox when they were young should get the Shingrix vaccine. So this is a fairly new vaccine. It's only been around for about 2 or 3 years. It replaced the older Zostavax. This only worked about 50% of the time. The Shingrix works with greater than 90% effectiveness. And if somebody does actually reactivate as shingles, it's usually very mild. So they're not likely to get that post-herpetic neuralgia. It's also an inactivated virus. It's recommended for people 50 and older. So ask your parents Christmas time, have you had that Shingrix vaccine? I got mine this summer, so that tells you how old I am. And the first dose was fine. The second dose really knocked me back. I had just terrible body aches just for a couple hours, and I took some Tylenol and I was okay. But it does have, you know, oftentimes you get a low-grade fever with that second vaccine. But for me, the Shingrix vaccine was even worse than COVID vaccine. So I got a lot of shots this year. So just again, encourage or you know, it'd be interesting conversation. We think that the Shingrix protection lasts at least 4 years, maybe better than that. It's new enough that we're not really sure. Again, a little side effects associated with that. Really, the only treatment we have for Hhv4 is acyclovir, just to try to lessen the pain associated with shingles or maybe the itching, severe cases of chickenpox. I had chickenpox inside my mouth and in my throat. So when that vaccine came out, I was like, kids, you're getting this one. let's avoid this. All right, so one more herpes virus I want to mention kind of briefly here. Hhv4, also called Epstein-Barr virus. This can be manifested in several different ways, depending on the immunological status of the host and where the host lives, as best we can guess. So one of the things that it's associated with is Briquettes Lymphoma. Your book calls it Briquettes Lymphoma, and when I was searching around on the internet, I think they just refer to it as Briquette Lymphoma more commonly. This was the first virus that was shown to be oncogenic. Okay, and it causes a cancer of the jaw. Epstein-Barr virus, well, I'll get to that in a minute. It's a very fast growing cancer. Also is associated with abdominal tumors. And kind of interestingly enough, it's most prevalent in African boys. Okay, in the US, we only have about 300 cases of this a year. And again, it's mostly boys. I'm not sure why that is, but it's much more prevalent in Africa, sub-Saharan Africa. And the best that we can guess of why that is that we have a lot of malaria in sub-Saharan Africa. So we think that the malaria kind of beats down kids' immune systems a little bit and makes them more susceptible to this virus. That's our best guess. And kind of interesting, this doesn't go latent in nerve cells. We have latency in B cells. The good news is this is a very treatable cancer. It responds really well to chemotherapy. So as nasty as this looks, maybe a lot of kids don't have access to chemotherapy in Africa, but at least in this country, it's very treatable. Epstein-Barr virus can also manifest as mononucleosis. So this is something that's probably more familiar to a lot of you, sometimes called the kissing disease because it's transmitted by saliva. So you usually need to have direct contact with that saliva, either kissing, shared water bottles, shared utensils, shared drinking glasses. So teams, keep your water bottles separate from each other. It's most common in teenagers and young adults. Very rarely will have a kid or somebody my age getting mononucleosis. The symptoms there, really sore throat, a little kind of a different sore throat than strep throat though. It's kind of almost like it feels swelled up, if I remember right. Just fatigue, it's varying severity of this. Like I had it for about a week, but some people will be tired just for a month or 6 weeks or even more than that. Enlarged spleen and liver. I've heard mono described as B cells fighting with the body's T cells. All right, so it's going latent in the B cells and it's kind of waging a war there and makes the host really super tired. So if you look at these different manifestations of Epstein-Barr virus, again, we think it's hypothesized that how it manifests has something to do with the host's cellular immunity. So for example, in this country, a kid with kind of a normal immune response when he's exposed to Epstein-Barr virus is not likely to have any symptoms at all. So some of us that were exposed as kids, you might be carrying that virus to some extent, might pass it on to somebody else, but you don't get sick yourself. Somebody that's exposed for the first time as a teenager or young adult would have that's in kind of their prime. You all are in your prime in terms of immunological status. You would have so much of an immune response that inflammation is kind of what causes the disease in this case. So you might have infectious mononucleosis. somebody like a child that lives in a poorer country that had a poor immune response, that's when, you know, again, maybe has something to do with malaria, might get the Briquettes Lymphoma. And then somebody that has almost no immune response, it's a lot more complicated than this, but we think then that can manifest as oral hairy leukoplakia, which are these kind of whitish lesions on the tongue. Sometimes they get hairy, not precancerous though. This usually clears up after a week or so. This is typically AIDS patients where Epstein-Barr virus would manifest that way. So we have a lot to learn about that, but that's kind of the hypotheses right now. All right, so other questions? Okay, let's do a few more review questions and then maybe we'll just start a little bit the next chapter. Okay. All right, sample of fluid from the lungs contains microbes that grow in filaments or clumps. That's your first clue. The cells stain poorly in the gram stain are red-pink when we acid-fast stain them. The bacteria in the sample are, all right, hopefully that's kind of an easy one, especially if you're in lab, taking your lab exam this week. That's going to be Mycobacterium species. Mycoplasma kind of sounds like Mycobacterium. These are cells that have no cell wall. And we maybe only mentioned that once, so I wouldn't really expect you to know that. They grow in filaments or clumps due to what virulence factor? Okay, cord factor. That's really the only two virulence factors that we mentioned for Mycobacterium: acid-fast cell wall and that cord factor that somehow is important for virulence. Okay, so acid-fast cell wall, mycolic acid, lipidy cell wall. Just kind of a tough one. I'll let you read this. See if you can narrow it down to a couple. All right, pathogenicity of primary tuberculosis. So first of all, cross out this one, form a pseudomembrane. That's actually diphtheria that does that. So again, this was not a question that I wrote, but if it doesn't sound like anything we ever talked about, then it's probably not the right answer. They do not destroy helper T cells. They are carried by macrophages to a variety of sites, but I think the question is referring to how does it get established in the first place? So this is not the best answer here. And then the correct answer here is that answer B. They prevent fusion of the lysosome with the vesicles. So Mycobacterium, remember, it wants to live inside macrophages. So it gets phagocytized, but then it has, ways to kind of outwit being killed when the lysosomes fuse. All right, immunization for what may reduce the incidence of otitis media and sinusitis? All right, so I gave you a little hint. The vaccine is called Prevnar 13. Okay, I don't think we talked about the name of the Bacillus vaccine or the Anthrax vaccine. We just mentioned that there was one. The Neisseria meningitidis is called meningococcal quadrivalent. That was the vaccine for that one. And the TB vaccine, BCG, Bacille Helmet-Guerin. That was the TB. So Prevnar 13, that's the strep pneumonia. So while the goal there is to cut down on meningitis, really, it's also going to cut down on ear infections and sinusitis. Okay, let's go ahead. We've got some little time here. Let's go ahead and at least start chapter chapter 25, and then we will have less to do next time. I'm not going to add more stuff just because we have time. All right, so I picked out 3 RNA viruses. There's a ton of them. There's a lot more RNA viruses that cause disease than DNA viruses. So I picked out Orthomixovirus, which causes influenza. We're also next time going to talk about HIV. And of course, we have to talk about coronavirus, and we'll try to talk about some of the, especially some of the updated information with some of these new drugs that are coming along. So stay tuned for that. We have worried, really, infectious disease specialists have worried for a long time that a pandemic was coming. But we thought, they don't include me as being an expert, they thought that it would certainly be influenza. So we were right, a pandemic was coming, and we're just wrong as to what would cause it. So we'll talk a little bit about that pandemic of 1918, 1919. That was caused, again, by Orthomixovirus. At that time, I cannot spell today. At that time, we didn't even know what was causing flu. We had guesses. We thought it was a bacteria that was called Haemophilus influenzae because we thought that was what was causing it. That was a time before electron microscopes. So this is a causative agent of influenza. Influenza is respiratory. So when I hear somebody say, oh, I had the flu, I was thrown up all night, that just makes me cringe, okay? You can call it stomach flu if you have to, but even really that is a misnomer, okay? So influenza is respiratory, you're coughing, you got body aches, you got fever, and I'll mention those again later. So probably people look at you sideways if you said, oh, I had enterocolitis, but that's the correct term for like throwing up and diarrhea. All right, so influenza, there are lots of different genera in this group, Orthomixoviridae. The ones that cause influenza are influenza virus A, influenza virus B, and influenza virus C. So the A is usually the most severe form of flu. B is usually a little bit milder, and C sometimes is asymptomatic or very mild disease. All right, so let's see, how do we describe these viruses? Once again, they're enveloped. You can see that over here from this graphic. Their genome is negative sense single-stranded RNA. So remember the difference between positive sense RNA and negative sense RNA. Positive sense RNA is in a form that can be read and recognized at the ribosome and translated into proteins. Negative sense RNA, you have to copy that into the positive sense, and then that can be read at the ribosomes. Okay, and that genome, it's segmented. So you can see in this picture over here, these little kind of curly cues. Those are the genes. Orthomixoviridae either has 7 or 8 segments. I think A and B have 8 and C only has 7 for some reason. So one hallmark of RNA viruses is that they have a lot of mutations. So they have to use an enzyme. Any RNA viruses has to be able to produce their own enzyme, an RNA dependent RNA polymerase, because we never make RNA from RNA in our bodies, right? So they have to bring in an enzyme that makes RNA from RNA, RNA dependent RNA polymerase, and those enzymes just typically make lots of errors. Okay, so they are typically going to have a pretty high mutation rate. But if the genome is in segments like this, then if something goes wrong with one of those genes and it doesn't get translated properly, transcribed and translated properly, then maybe the other segments would still be able to lead to a viable virion. So it's maybe an advantage of having segments versus one linear piece of nucleic acid. All right, another hallmark influenza virus is, like lots of viruses, they have glycoprotein spikes. Okay, so there's two different kinds. Hemagglutinin Okay, I spelled that right. So usually abbreviate that H. The hemagglutinin spikes are necessary for attachment and fusion. Okay, so that's the big red ones here on the picture. And then the second spike, neuraminidase. Sorry, neuraminidase. Okay, so usually abbreviate that H or N, H and N. Neuraminidase helps a little bit apparently with the fusion of the virus to the host cell, but is most important in virion release. So remember, we correctly call something a virion if it's got like the envelope and the capsid and the nucleic acid all together. Technically, the virus is just the nucleic acid broken away from the rest of the parts once it's inside the host cell. Okay, so we name the strains based on the subtypes of hemagglutinin and neuraminidase. So there's actually 16 different types or subtypes of hemagglutinin and 9 different subtypes of neuraminidase. Okay, and not all of those subtypes are forms that are able to infect humans. But again, this is how we name strains, like H1N1. We also called that the swine flu. Okay, that's one that's able to infect humans. And I thought it might be helpful to talk about how we name the strains of flu. Because there is so much mutation, these strains are always changing. It's kind of a slow change. We'll talk about that antigenic variation in just a minute. But again, they're constantly changing over periods of months, usually. So here's a couple of examples of names of strains. The A, that stands for what type, whether it's influenza A, B, or C. The B and C are ones that only infect humans, but A can infect some animals as well. So those tend to be more severe because we get some reassortment. We'll talk about that in a second. The source of the virus, horse, pig, where it was first isolated, where it was first found, the year, this I think is just a subtype there, that number, the year that it was found, and then here's the hemagglutinin and neuraminidase forms. So H3N8 or H1N1. Okay, so let me finish with just talking about this genetic variability. So we talked about Neisseria gonorrhoeae, how it's really good at antigenic variation. It has mechanisms to switch up the amino acids on its pelin. That's kind of the most antigenic part of it, the outside of the cell, the pili, sorry. Well, pelin's a protein, so changes the amino acid sequence there, and that just eludes our antibodies. It eludes our immune system because it happens so fast within, a week or two. In flu, we have a couple different kinds of antigenic variation that can be a little bit slower. Antigenic drift, antigenic shift. Okay, so antigenic drift is when you have gradual point mutations over time in those spike proteins. Okay, so this happens maybe over a year or two. That's usually responsible for, we'll call it biennial flu spikes in cases, arise in cases. So for instance, usually 1 year we have a lot of flu because that's kind of the same strain that's circulating the next year. We might have less the next year. And then, it continues to drift. We can continue to have mutations and you have higher the other year. So if we look at data, flu data over the years, we tend to see this kind of every other year higher. A more abrupt change that can cause usually a little bit more severe epidemic is antigenic shift. And that's when we get reassortment of those H and N spike proteins in another animal. Okay, so we might have, this picture shows this better. If you have a human strain of flu and say a strain that only infects ducks, at the same time infecting a pig. So you get reassortment between those two strains and then come out with something totally novel, like H3N2 that wasn't in any of the original ones. So that's usually responsible for some strain that hasn't been seen before and that people don't have immunity to, and that can be more severe. All right, we'll pick up with that next time, and we're getting there, almost there. I'll be in my office for a while if you want to look over exam, anything like that. Have a good day.